Welcome to today's lecture on the intricate world of apolipoproteins and their roles in lipid metabolism. In the realm of biochemistry, understanding how lipoproteins function is crucial for comprehending various diseases and identifying potential therapeutic targets. In this comprehensive script, we will delve into 12 fundamental topics, ranging from the regulation of VLDL secretion to the implications of lipotoxicity and lipoprotein-related diseases. So, let's begin our journey into the fascinating world of lipoprotein metabolism. Apolipoproteins are proteins that bind to lipids to form molecular complexes called lipoproteins, which serve as the major transport vehicles for lipids in the blood. These proteins play crucial roles in lipid metabolism, including lipid transport, cellular uptake, and enzyme activation. There are several classes of apolipoproteins, such as APOA, APOB, and AP, each with distinct functions. For instance, APOA1 is the major constituent of high-density lipoproteins, HDL, and is involved in reverse cholesterol transport, while APOB is present in low-density lipoproteins, LDL, and very low-density lipoproteins, VLDL, responsible for transporting cholesterol and triglycerides from the liver to peripheral tissues. Very low-density lipoproteins, VLDL, are assembled and secreted by the liver and play a crucial role in carrying triglycerides to adipose tissue. The regulation of VLDL secretion is a complex process influenced by various factors, including insulin, free fatty acids, and genetic variations. Insulin stimulates VLDL production through the activation of sterol regulatory element binding protein 1C, SREP1C, and inhibition of VLDL catabolism, contributing to elevated triglyceride levels observed in conditions such as obesity and insulin resistance. On the other hand, free fatty acids enhance VLDL production by promoting hepatic lipid synthesis. Moreover, genetic variants in genes involved in VLDL metabolism, such as apolipoprotein E, APOE, and microsomol triglyceride transfer protein, MTTP, can influence VLDL secretion rates. Acyl coenzyme cholesterol acyltransferase, ACAT, is an enzyme present in various tissues, including the liver and macrophages. Its primary function is to esterify cholesterol, converting it into cholesterol esters, CE, which are then stored within cells or packaged into lipoproteins for transport. ACAT has two isoforms, ACAT1 and ACAT2, with distinct tissue distributions and roles. ACAT1 is primarily involved in cholesterol esterification within macrophages, regulating cholesterol accumulation in atherosclerotic lesions. ACAT2, on the other hand, is essential for cholesterol esterification in the liver, contributing to the production of VLDL and the regulation of plasma cholesterol levels. Beyond its structural role in cell membranes, cholesterol also plays a crucial role in cell signaling. Cholesterol-rich microdomains, known as lipid rafts, serve as platforms for the recruitment and activation of signaling molecules. These lipid rafts facilitate receptor clustering, optimize signaling efficiency, and regulate the spatial organization of signaling pathways. Additionally, cholesterol can modulate the activity of membrane-bound receptors, including G-protein-coupled receptors, GPCRs, and receptor tyrosine kinases, RTKs, by altering their localization and function within the plasma membrane. Dysregulation of cholesterol metabolism and disturbances in lipid rafts can have profound implications for cellular signaling and contribute to various diseases, such as cancer and neurodegenerative disorders. Scavenger receptors are a class of cell surface receptors primarily expressed in macrophages and endothelial cells. These receptors play a critical role in the clearance of modified lipoprotein remnants, cellular debris, and pathogens. Scavenger receptors, such as SRA, CD36, and LOX1, recognize and internalize lipoprotein particles containing modified lipids and apolipoproteins, particularly oxidized LDL, oxaldol. This process leads to foam cell formation, a hallmark of atherosclerosis, contributing to the progression of cardiovascular diseases. Understanding the mechanisms underlying scavenger receptor-mediated lipoprotein metabolism is crucial for developing therapeutic strategies aimed at reducing atherosclerotic plaque formation. Lipoprotein A, LPA, is a lipoprotein particle composed of an LDL-like particle covalently bound to a unique protein called apolipoprotein A, ApoA. Elevated levels of LPA are associated with an increased risk of cardiovascular diseases, including coronary artery disease and stroke. The proatherogenic properties of LPA can be attributed to various mechanisms, including the promotion of oxaldol formation, inhibition of plasminogen activation, and activation of inflammatory pathways. Furthermore, genetic variations in the LPA gene, encoding apolipoprotein A, contribute to interindividual differences in LPA levels. Understanding the biology of LPA and its role in atherosclerosis is critical for designing effective therapies for cardiovascular risk reduction. ATP binding cassette, ABC, transporters are a superfamily of transmembrane proteins responsible for the transport of lipids across cellular membranes. Several ABC transporters, including ABCA1 and ABCG1, play key roles in cholesterol efflux, a process by which excess cholesterol is removed from cells, particularly macrophages, and transported back to the liver for excretion. Dysfunction of ABC transporters can lead to impaired cholesterol efflux, resulting in the accumulation of cholesterol within cells, foam cell formation, and the development of atherosclerosis. Investigating the mechanisms governing cholesterol efflux and its regulation by ABC transporters holds promise for the development of novel therapeutic interventions for cholesterol-related disorders. Lipoprotein lipase, LPL, is an enzyme critical for the hydrolysis of triglycerides present in circulating lipoproteins, such as chylomicrons and VLDL. LPL deficiency leads to familial chylomicronemia syndrome, FCS, characterized by severe hypertriglyceridemia due to impaired lipoprotein clearance. The consequence of reduced LPL activity is the accumulation of chylomicrons and their remnants, resulting in symptoms such as abdominal pain, eruptive xanthomas, and pancreatitis. Understanding the molecular basis of LPL deficiency and the pathways involved in chylomicron metabolism is essential for early diagnosis, management, and potential therapeutic approaches for individuals affected by this disorder. Macrophages play a central role in the development and progression of atherosclerosis, a chronic inflammatory disease characterized by the accumulation of cholesterol-rich foam cells within arterial walls. Cholesterol trafficking in macrophages is carefully regulated to maintain cellular cholesterol homeostasis. Uptake of oxalate via scavenger receptors leads to the esterification of cholesterol by a CAT and subsequent formation of lipid droplets. Cholesterol efflux from macrophages, mediated primarily by ABC transporters, is crucial for preventing cholesterol accumulation and foam cell formation. Dysregulation of cholesterol trafficking pathways can disrupt macrophage function and contribute to the inflammatory milieu in atherosclerosis. Lysosomal acid lipase, LAL, is a critical enzyme responsible for the hydrolysis of cholesterol esters and triglycerides within the lysosomes. Deficiency of LAL activity leads to lipid storage disorders, such as Wolman disease and cholesterol ester storage disease, CESD, characterized by the accumulation of cholesterol esters and triglycerides in various organs. These disorders manifest with hepatosplenomegaly, liver dysfunction, and cardiovascular complications. The understanding of LAL function and the pathogenesis of lipid storage disorders is essential for developing targeted therapies, including enzyme replacement therapy and gene therapy, to improve the prognosis and quality of life for affected individuals. Genetic variations in genes involved in lipoprotein metabolism can significantly impact lipid levels and the risk of cardiovascular diseases. Several well-known genetic variants have been identified, including those in genes coding for apolipoproteins, AP, APOB, enzymes, LPL, ACAT, and regulatory proteins, PCSK9. For example, the AP genotype influences plasma cholesterol levels, with the AP4 variant associated with elevated LDL cholesterol and an increased risk of cardiovascular disease. Understanding the genetic underpinnings of lipid-related traits can help identify individuals at higher risk, guide personalized prevention strategies, and potentially reveal novel therapeutic targets. Lipotoxicity refers to the harmful effects resulting from the accumulation of excessive lipids, particularly fatty acids, in non-adipose tissues. Dysregulation of lipid metabolism can lead to lipotoxicity, contributing to the development of various diseases, including metabolic syndrome, non-alcoholic fatty liver disease, NAFLD, and type 2 diabetes. Lipoproteins, especially LDL and VLDL, have been implicated in the pathogenesis of lipotoxicity. Oxidized LDL, for instance, can induce cellular oxidative stress and inflammation, promoting the progression of atherosclerosis. Understanding the mechanisms linking lipoproteins, lipid accumulation, and lipotoxicity is crucial for developing effective interventions to mitigate the burden of lipoprotein-related diseases. In conclusion, this script has provided a comprehensive overview of various aspects of lipoprotein metabolism and their implications in health and disease. From understanding the roles of apolipoproteins and lipid transport to exploring the consequences of genetic variations and lipotoxicity, it is evident that the intricate network of lipoprotein metabolism is essential for maintaining cellular homeostasis. By unraveling the complexities of lipoprotein metabolism, we can not only enhance our understanding of diseases such as atherosclerosis and lipid storage disorders but also pave the way for innovative therapeutic interventions aimed at preventing and treating these conditions.